Cool. Well, I really appreciate you joining us again. This is such an interesting subject and something that I still have a very amateur perspective and knowledge level about. And really appreciated all the information you had for us last time, and looking forward to what you got to share this time, and hoping to get some good questions for you too.
Perfect. Yeah. Yeah. I am glad I shared last time. I just love talking about this to people, and I think it's good for people who are interested in especially history and linguistics to understand this sort of genetic stuff as well. I do think I was a little bit underprepared last time. I just wasn't exactly sure what I was going to talk about and whatnot. So I have a better slideshow this time, basically, is the long and short of it. I
Great.
hope it's a little bit easier to follow for everyone. But anyway, yeah, I guess should I start sharing it
Yeah.
basically?
And you are co-host, so you should have all those permissions and everything.
Okay, let me know when you can see it.
I see it perfectly.
Okay, so last time I talked more about, even though it's actually somewhat less used, although that's somewhat changing, but basically last time I talked more about like haplotype based methods, essentially. I kind of give like a vague overview of them. Sort of like that's kind of my main focus, which is like when you have kind of long tracks of shared DNA. But actually in ancient DNA, we mostly have been using allele frequency based methods. Which yeah, instead of using like the kind of long sharing, you kind of look at like an average over all of the sort of allele frequencies over the whole genome, which I will get into explaining what allele frequencies are and yada yada. I'm kind of saying a lot of jargon, but basically you are kind of looking at every individual locus. It's with some level, and then kind of averaging over the whole genome, and instead of like looking at like, oh, these like long tracks are identical, which is kind of more what I was talking about last time. And there's a lot of advantages to this. When done right, there's like you can kind of have it be like an unbiased estimate. And so like you're not like haplotype based methods, there's a like population size has like a large effect, where you can kind of isolate more what you want with these statistics because they're a little bit simpler in some way. You also get p-values out of them really easily, which allows you to kind of reject hypotheses, and we'll talk about that kind of if we get deep enough into this talk. It's like 20 or so slides, maybe 25, but we don't need to get through all of it. I'll just yeah, we'll hopefully get through it all. And if we have more, I actually can do more because this is just basically stolen from my class that I helped teach, so I have more slides that I've made. Anyway, but basically, yes, there's a lot of advantages to them. And another big one is that, like with kind of like you need high quality genomes, and often depending on what you're doing, a lot of them, for these kind of haplotype based methods, where you're kind of again looking at these like long regions of sharing, you need to have like long, like continuous reads or whatever. But you have a lot of missingness. You can still actually do things with these type of methods. And with ancient DNA, especially up until recently, as we get like better and better coverage and more genomes, it's been a little bit unfeasible to do haplotype-based methods. So this has been kind of the main workhorse of ancient DNA, actually, are these statistics I'm going to get into today. I mean, that's just sort of intro what they are. And yeah, I don't know if I had anything else to say on that. But yeah, so this is like F3, F4 statistics. I don't know if you've, or D statistics. I don't know if you've heard any of these terms, but they're very common among the literature. And even other things that like, QPADM, again, I'm not sure if you've heard of it, but if you've read an ancient DNA paper, you might have. They're all based on this sort of thing. I will say recently it's been changing. We have like more and higher quality genomes, and we're also kind of looking at more fine detailed problems where it seems like allele frequencies are kind of struggling to like find any different. I mean, like these two populations are so similar, their frequencies of their alleles are so similar, it's almost impossible to distinguish them. And so there's been new methodologies. I will just shout out two papers, Speedo et al. And then also McCole et al. For people who are interested, because they both use interesting kind of new ways of kind of pushing the boundary with haplotype based methods. And also they're both preprints that came out this year, and they're both about the spread of or if they talk about the spread of Germanic languages. And so if people, I assume, are interested in that, which, given what your channel is, Jackson, I feel like people will be. I just wanted to put that out there, and I think that if people are interested, they should look for those papers. Anyway, I'll get to actual talk now. Sorry, it's kind of a long intro.
And do you have the full citation info for those papers that you can show?
Uh, I can grab it if you want. I don't have it
Maybe
off the top of my head
toward,
though.
toward the end. Yeah, maybe just something to leave people with, yeah.
Yeah, remind me and I'll grab it.
Great.
Uh, yeah, it's Hugh McColl and Leo Spiegel are the first authors respectively, and they're both 2024 papers, and they're both on bioRxiv. And I think you just look up like Germanic languages, but I will remind me, and I will get the actual like link. Okay. So I wanted to just start off with some basic PopGen terminology, which I will be using, and it's kind of jargony. So for people who are kind of totally out of their depth, I want to, if I'm saying things, I want people to understand them. Also, if I'm going too fast, people in the chat or Jackson or anyone, please let me know, and I will slow down. I get excited when I talk about this kind of stuff. So first, like what a base or a base pair is it's a single DNA nucleotide. It's like, you have like a long string of these, nucleotides, and they're all kind of chained together, and that's what forms the overall like DNA strand. And this can either be an A, a T, a G, or a C. That's called a base pair basically, and it's called a pair because there's two of them, right? So if you have like an A, you'll have a T on the other side. If you have a T, you'll have an A on the other side. If you have a G, you'll have a C, and G, you'll have a C. There's like a pairing there. Um, Anyway, so that's what a base pair is. And then a gene is sort of like a length of DNA, and it can be like as short as a base pair, but it's it generally but not always encodes information. It's kind of I feel like it's used very loosely, and all these terms are kind of not always used. Like different people use them somewhat differently. But generally, like a gene is something like usually codes for like a protein or like that's kind of it's usually like it's coding for something. And then sometimes like regions that are like affect transcription but aren't actually making the protein. Some are called genes, but they're anyway, it's kind of vague, but that's like what a gene is. It's like usually something that actually encodes for like something that's produced in the body or whatever that then goes on to form something. Um, and then a haplotype is kind of has like a similar meaning. It's like a long string, but there's less of an emphasis on like information, and like they can kind of overlap. Like a haplotype is just sort of like a like long string of characters, which like two people might share. It's like a yeah, it's like they have the similar type of like a haploid string. Haploid means that there's like this one copy of the DNA, where diploid is like you have two, which is humans are diploid, for example. We have a copy from our mom and a copy from our dad. Anyway, I'm kind of going out in the weeds. I should come back. So, okay, the next thing is what an allele is. So it's a variant of like a gene. And that can even just be like a variant at like a single SNP. And it doesn't necessarily need to be coding, but like that's what an allele is. It's a variant of like haplotype or a base pair or a gene. So it's a variation basically. And so I have three examples of alleles here. You can see that the second one compared to the first has a bunch of repeated T's. So there's kind of like an insertion there or like a duplication or something, but you can see that they're like mostly the same string, but there's like a difference. And then the third one, you can see that it goes AATCG. Compared to the first one, that's what the first one and the second one do, but that one then says it goes AATCA. So there's like a single base pair difference, basically. Um, and so that's like an allele, basically. And all three of those are like different alleles. You have like three different alleles. And so it's like, oh, what allele do I have? I have the allele that has like a bunch of T's repeated, right? That's like a thing I could talk about. It's like which of the possible variations of the possible variants do you have? And then often people will say like a gene copy or a allele copy to refer to like the physical copy because like I guess I have like a copy of that allele, right? Like I have a copy of that. It's not like I have like the kind of the like one of five T's is kind of maybe more of like a metaphysical, like it's like a it's a thing that you could have, right? And if you have one, you have like a copy of that. That's not the most used, but I think it's important to put out there. This is also our stuff. I might not talk about all of this, but these are things that I think are useful terminology for people who want to read DNA papers and read about population genetics in general. Okay, and then the thing I will be talking about a lot is a SNP, which is an SNP, but people just call it a SNP. And it stands for single nucleotide polymorphism. So that's kind of jargony again, but we'll kind of go through it, break it down. Get nucleotide, it's like a base pair, it's a single thing of the DNA, and single, it's like a single one. So it's just a single base pair. And polymorphism is like poly is many, and morphism is like form. I think my etymology is not the best, but it's like there's multiple things it could take, right? So it basically means that there are multiple alleles. So there exists some variation, i.e., more than one allele is present at this SNP. So the population has, they can have a T, they can have a C, maybe they can have a G. It doesn't really matter. As I'll kind of probably double back on, we usually assume things are biallelic, but they don't have to be. You can have all four possible variants. You can have A, T, C, and G all at this site, at which point it would be a like quadrillelic, I guess, SNP. And these are literally the bread and butter of all allele frequency methods. You're kind of averaging over all SNPs. So you look at like actually most of the human genome doesn't vary at all. Like one around average 1 in 1,000 sites between any two people will be different at all. And so yeah, basically you yeah, so you can ignore the other like 999 allele. Sorry, the other 999 loci. You can ignore all the other ones except for the one that is a SNP. So, all right, is that all making sense, by the way? I want to not go too fast.
I think so.
Okay. I realize I speak fast and I get excited, so I'm trying to rein myself in. But um, anyway, so continue on terminology. So the allele frequency is a number between 0 and 1 that represents how common an allele is in the population. So, you can have the frequency of, say, the T allele at some locus. I think I didn't put in locus. I should say that is, though. A locus is like a single location, just it's like a, some part, some base pair in the genome. So, at some locus, you have 30% of the population has an A and the other 70% has a T. And so like that's just like a possible thing you have, at which point the allele frequency of the A allele is like 30%. So this one maybe is more standard, but mutation, if people are not familiar with it, is the process by which a new allele is created, right? So the new variant is created. And because we're going to be working with recent events like human history, we can assume no double mutations. And so basically, if the mutation rate is so low that actually if you look at humans and chimps, basically the like the number of only 1 in 100. Do you see like the like both humans and chimps have like the same variance? You can almost just like if you have a C and a chimp has a C, you can almost always bet that like you have a common ancestor that had a C with the chimp because only like that time scale 5 million years, only like 1 in 100 loci will have been hit by two mutations that like did the same thing. So if you have two mutations, like if you have a shared, like if you have a shared allele, you can almost always guarantee it's because of a shared mutation event, not because of like two different ones that produce the same allele. And so this is often called like the infinite like alleles model, basically, where like you can or the infinite size model. I mean, the point is you can basically yeah, you can ignore double mutations. So we're gonna just pretend that every time you get a mutation, it's always new, always novel.
That makes
Uh,
sense to me. And it's similar to linguistics where you look at two languages that are otherwise clearly closely related. If they share a feature, it's more likely to have been shared by the common ancestor than that they both independently innovated it. Yeah, I see what you mean.
yeah, I think there's I think a lot of this people familiar with linguistics, especially like the tree stuff, it'll be very familiar. I think there's a lot of sharing there. Um, I'm not a linguist. I think it's even maybe more extreme than linguistics, though. And I'd love to hear your thoughts on this because I feel like with linguistics, my understanding is that like you do see sort of weird like double features, like like false friendling is the term, and also just like similar sound shifts in two different languages that like are opposites, like W to V, like it happened multiple times in different like in Latin and also Old Norse, and then like you have like bad meaning bad in both like I think Persian and English is like the classic example, and those aren't shared. So I think it does happen occasionally in languages, right? Or is that am I kind of incorrect there?
Well, but those are fairly distant from each other. I mean, like if you look at something like the in both English and the Scandinavian languages, the typical way of forming the past tense of a verb is adding, a D or a T suffix. But if you have something like sit and the past tense is sat, and that's the same in both English and Icelandic, that strange feature in that one word is more likely to be inherited from their common ancestor when we otherwise know those two words are those two languages are closely related.
That makes sense. Yeah, I guess you just can think of like the time scale of genes as being like just infinitely like not infinitely, but vastly larger. So like well like the level. I mean, this is I'm not sure this comparison is accurate, but the level of relatedness between like English and Icelandic is like the same as like humans and chimps, even though like vast time differences. Yeah, basically just it's a very slow process is the main
I,
point. But that makes
I
sense.
yeah, I'm just trying to analogize it to something that I know a little bit better and test my understanding of it. But I think we're talking sense to each other.
Yeah, no, I think it's great. I think we should. I think that's good to do. I think good for you, and I also think actually having more analogies will be useful for people who view it on YouTube and here. And also, I think that I think probably more of your audience probably has a linguistics background than a genetics background. So I think it's definitely very good to do. Um, anyway, yeah. So hopefully that analogy is accurate. I think it is. I'm not enough of a linguist to say, but I think it is. I think it is. And then yeah, recombination next is just the way that like it's the way that like the you have a long chromosome from your parents, and then you inherit like you give it to your offspring, and it will be broken up basically. So you have one copy from your mom, you have one copy from your dad, oh, so, yeah, mom and dad, and then there's randomly kind of breaks apart and reshuffles. I think we talked about this a little bit last time as well, and it's the way variants are shifted and kind of spread across the population. Maybe you could make an analogy here with like linguistic borrowing, although I think that's not really actually the right thing here. But anyway, but basically it's just like the shuffling up of the genome. So if you have a mutation here and if you have a mutation kind of nearby, they come into being in like similar locations. In the future, you can either have one or the other and not both. But if they are close, you're more likely to have both because it's not instantaneous to get broken apart. But over time, these things will be kind of broken apart. And linkage sort of is how linked two alleles are, and it's basically how like much it is not been broken up by recombination. So if I have like some gene at a locus and then another, or not some gene locus, some allele at a locus or whatever, and I have another one that's like 100 base pairs away, very, very close, they're probably highly linked. So if you have one, if I like know about my DNA, I have an A here, then I can almost guarantee I'll have like a C there because they're always together, basically. Versus if they're much further apart, then I don't know that as confidently. Um, and so then Morgans are like I think there's a guy named something Morgan who was a geneticist, and that's what's named after. And anyway, essentially they're a metric of genetic distance. So you could just count like the number of base pairs between, but actually this is often more useful in some way. And it's basically like a distance that's in terms of likelihood of recombination. I'm not getting to all the nitty gritty of it. It's not the most straightforward thing in my opinion, but basically a centimorgan, which is a hundredth of a Morgan, is the chance of there being like there's basically a 1% chance of being recombination if something is one centimorgan away. And so like the expected number of recombinations is like 0.01 essentially. And like a Morgan essentially is like if you're one Morgan away, essentially you're like the expected number of recombinations is one. It's not guaranteed to be one, it could be two or whatever. But like that's like the likelihood of recombination per generation as well. So if like there'll obviously be recombination as there's more and more generations. Okay. I'm gonna move on, but if please, Jackson or anyone in the chat, stop me if I'm ever going too fast. Okay, so let's set up to do some actual like an analysis and sort of a thought about this. We're gonna make some simplifying assumptions. There's like a great quote that's like, all models are wrong, but some are useful. So this is obviously wrong, but it's useful essentially. So we're going to assume that all SNPs are biallelic, and in humans this is like almost always true. There's occasionally a third of one, but it's like very rare. And we're also going to assume no double mutations. I think I already kind of mentioned this. Unless we're looking in selection, we're going to assume no selection. There's kind of an assumption that the genome is like mostly neutral. It's like often thought of as like being mostly junk DNA, and like basically a lot of it is not really under strong selection is the idea. And like the variation that we do have is also like it's like permitted variation, right? So if half of humans have allele one and other half have allele two, they can't be that bad because if you had like a terrible allele, then it would be like purge of the population pretty quickly. So when we're keeping track of alleles, actually doesn't really matter so much. But we need to like decide which one is like a zero, which one's a two. That'll make more sense in a second. But we basically need to keep track of which allele is which, especially in like a mathematical way. We can't just say ATGC because then we can't do the math we want to do on it. And so we need to decide which one is like an X and which one's the Y. So the way we normally do this is we say that one allele is ancestral and one is derived. And there's a very simple way of doing this, which is just you look at the chimp and you say, what does the chimp have? And you just can always assume that is the ancestral allele. And you'll be right 99% of the time. And being right isn't always the most important, at least for most analyses within humans. It can be, but for most analyses, it's not that important. And we only get it wrong like 1% of the time. So this is a very simple thing we can do. And if we want to be more confident, we can also look at like gorilla and orangutan and other things like that. But the reason we can just look at chimp isn't because chimps are like less evolved. I actually think there might be more differences. I could be wrong about this, but I remember reading something like chimps have more differences since the last common ancestor than humans have differences since the last common ancestor somehow. Again, I'm not sure how it was calculated, but that is something I vaguely remember hearing. And the point is that either way, chimps are not like less evolved. The thing is that because there's no double mutations, if chimps don't have a variant and humans do have a variant, we can assume the mutation was on the human side. So the mutation must be like a new thing in humans, and so that must be the derived, the like new state. And you actually can do the exact same thing with chimps. If you want to look at chimp genetics and you were like flipping it around and I were only interested in like internal chimp variation and I wanted to find what the ancestral variant was, I would just get humans. And whatever one, like if chimps have two different alleles and humans only have one of those, then that must be the ancestral one, whatever humans have. And again, you get it right 99% of the time by doing that. So anyway,
I,
it just is useful to have. Oh, sorry.
I see what you're saying. Yeah, it's, I mean, again, on a language analogy, two languages that share a feature, that is going, you can treat that as the original trait. When they differ from one another, obviously it is derived, it is the derived one with respect to the ancestral state. It doesn't mean that both haven't changed, but it means that when both have something that's the same, that's older.
Exactly, exactly. And I think to actually to build on that, you can think of like when there's variation in humans or like variation in chimps, it doesn't really matter, but there's variations at like a locus, there are two alleles. You think of it as like two dialects in of the same language. And you say like there's two dialects of the same language, and there's a different related language over there. And you can see which dialect has the feature like the other language and say that is probably the ancestral state. And the dialect that differs from the neighboring language, like the related language, is the derived state.
Yeah, this makes sense to me. Also,
Yeah.
this is making me really curious about comparative evolution of chimpanzees and humans now, but that's not what we came here to talk about. So,
Yeah, it's out of my wheelhouse for sure. It's not something that I focus on, but because I do go to like talks on, genetics, I occasionally hear this sort of thing and like, yeah, there's like all sorts of interesting things about like which regions of the genome are selected in humans, and then there's also like selection in chimps, like you can kind of use that to like, humans always have like a lot of selection on our brains. I think it's like a not surprising thing. We also have a lot on our stomachs, like which anyway, I don't know. There's all sorts of interesting things to think about, like what the ancestor had. But again, not for today, and definitely I feel like I'm not the right person to talk about it either. Um, okay, so yeah, so and I kind of mentioned this already, but we basically just use ancestral drive to keep track of allele frequencies, right? So if you say that way, instead of saying, oh, it's like allele frequency of 0.3a, we can just say there's allele frequency of 0.3, and we know that that means that it's like 30% derived and 70% ancestral. So the ancestral derived thing is just there to keep track of things. Okay, so now we're gonna do some basic pop gen. We can skip over this, but I thought this maybe would be a good thing to have your chat or you do if you guys want, just to like, I don't know, kind of ingrain it in your head a little bit more, I suppose. It's not actually that important for later stuff. Anyway, basically, we're going to call the ancestral allele Q and the derived allele P. And so if you have an allele frequency of 0.25, that means 1/4 of the gene copies in the gene pool are P and 3/4 are Q. So it means 25% of your like of the population is derived for the allele. So, okay. So again, we can skip this if I don't know if you want to, but like this is also, like I said, it's kind of taken from a class that I was TAing, so that's why it has like some of the stuff. And I considered deleting it, but I actually decided I would leave it as a skippable thing. But so if we have like an allele frequency of 0.4, like what percent of individuals are heterozygotes? And I'll kind of at least do one of these to just kind of walk the whole chat through, or like the whole Zoom through, and then if people want, we can do more. And then so what percent are homozygous for the P, and which percent are homozygous for the Q? And so basically every human has two copies, right? And if we assume that like there's no structure, there's no like internal distribution of some people have more of P and some people have more of Q. There's a 40% chance that either of your allele copies will be the P version. So the chance of you having two P's is just 0.4 times 0.4, which if I'm doing my math in my head correct is 0.16. So 16% of people will have both P's. They'll be identical P squared. And then I kind of did it in the wrong order, but now going to the heterozygotes, which heterozygote means you have one of each. So you have one P and one Q, like I say there. So that means you have a 40% chance of having that times a 60% chance of having the other. So that's 24%. However, you need to double it because it's sort of weird, but you can have a P and a Q, or you can have a Q and a P. So this sort of like the order can be flipped. And so it's actually 2 times P times Q. So it's that doesn't make sense. I can kind of go through more details, but like, you basically have one, like if you have a P, you can then have a Q. But if you got a Q the first time, then you can get a P the second time. Kind of you're like dealing out these things one at a time, right? So if you get deal the P, then you need to get a Q the next time. To get a Q, then you need to get a P the next time. So essentially, it's 2 times 40% times 60%. So I think I said that was 24 if you just did P times Q. So it's now 48% is going to be are going to be heterozygotes. Almost half of all the people will have one of each. And
And
then oh,
and these heterozygote situations, there can be a difference depending, like if P and Q are on the paternal and maternal side, that can be different from if P and Q are on the maternal and paternal. Is that, or am I
um,
on you right?
I think I didn't say it very clearly. Like I guess I was just saying that you can kind of like you count it like I'm not counting them differently, but I'm like if you kind of did this and you sort of just did it really intuitively and speedily and didn't think very deeply about it, you might say, oh, the heterozygotes, you have to have one P and one Q, so it's P times Q, and so it's 24% of people have that. But it's actually double because you do have like the one case for mother and father, and then one place for father, mother, like yeah, like so there is a difference just in like when it's not like they like would be genetically different. Like they'd be kind of indistinguishable unless you kind of knew who the mother and father was. But it's just like a sort of difference in the way you calculate the heterozygote versus calculating the homozygote. Because the homozygote you have to get one from the father and one from the mother. There's only one option, right? You can only have it on both. Well, like you kind of said, there's kind of two options with the heterozygotes. You can have one from your mother, one from your father, or the reversed. And because
Hmm.
there's two options, you need to double the rate. Also, someone in the chat is saying something.
Yeah,
Does
Blake
that
said something much like that.
Yeah, exactly. Yeah. So the Blake is saying something similar. Does that explain it well enough that time? I
Yeah,
may not.
yeah. No, I think I'm following you.
Okay, perfect. Yeah, and then last but not least, we have the other homozygote, the homozygote for Q, which is zero point 0.6 times zero point 0.6, so just 0.36. And if I did everything right, I'm not going to do this out in my head, but like, it all should sum to 1. So like 0.16 plus people with a calculator in front of them can check me, plus 0.48 plus 0.36 should be 100% because the whole population. So if anyone wants to, we can do this is really up to you, Jackson. People can try figuring out what they would be if the low frequency is 0.1 or 0.7. Otherwise, we can move on. I mean, this can be like a fun. People on YouTube can pause it here and whatever and play around with themselves. But I don't know. I just thought I'd keep it, but we can definitely skip it.
It can be an exercise for home, unless there's someone, hey, you're here right now. Anybody feel like running through this?
I'll give like 2 seconds to the chat and then we'll move on. Um,
Okay, Blake has answers.
and Blake is right for the first one. The first one, Blake is correct. I won't reveal it for people on YouTube who maybe are feeling excited to do this.
Do an answer key to
Wait,
the
maybe I should. Yeah. So Blake says 0.01, 0.18, and 0.81 for the first one, which is correct. So
okay, yeah, just working backwards from it's backwards from the way the first question is phrased.
yeah. And then we have an answer for the second one, which is also correct. And yeah, so it's 0.49, 0.42, and 0.09. And then yeah, I guess I'll go through the next two just kind of vaguely. I'm not even give the answers, but I'll just kind of go through them very quickly. Because I think that they're kind of interesting and like they're kind of different than the first one, as you can tell. So basically, the point of the first one is that if everyone mates randomly, you have an even like sorting of these variants. So you have 10 you have 15 with PP, you have 10 with PQ and 20 with QQ. And so you don't have like the sort of ratios that like the ones that we gave and the ones that Blake gave in the chat, which are like these like kind of Hardy-Weinberg equilibrium where you have like you have exactly P squared of the PPs and you have like Q squared of the QQs. There's like very nice kind of formulaic thing and 2 PQ of the PQs. So you don't have that to start off with, but with one generation of mating randomly, you do end up with that, which is kind of an interesting phenomenon that like if you have one generation of random mating, you'll end up with this distribution. Um, okay, and then if you have a population of 70 individuals, this one's maybe more important for our purposes, at least what we're going to get, where basically 70 individuals with a frequency of 0.1 are then joined by 10 new individuals who their low frequency of this locus is 0.9. And so basically when you do this, you now have to do like a kind of a weighted average of the to get the new allele frequency. So you have like what you would do is you'd figure out what the allele frequency is in the population, which we haven't already given 0.1. So it's like, you can say that there's 70 individuals, it means 140 um gene copies, right? Because you have two for each person, so 140, and you say 10% of those or whatever are like P, so you have 14P, and I'm not sure. Let me do it. I might be getting this wrong. 126Q. And it's then joined by 10 people who have then nine more P and like one more Q. You then add that all together, and you divide, you like simplify or whatever, and you'll get some new percentage for the larger population, right? You'll have some new percentage for the actually, sorry, it's 10 minus, so it'd be 18 and 2 for the exact reasons I said before. Where it's you have to double it because it's two people. Anyway, there's two copies per person. But the point is that you then will end up, this new population will then end up with a allele frequency in between the first two, right? And so this is like what happens when you have admixture between two groups. So like, when the Old Norse go and they capture people from Ireland or whatever, and they bring them to Iceland, the allele frequency of any given variant, if this is true, we know that Icelandic people do have ancestry both from like Scandinavia and from the British Isles. People of Iceland have, any given variant. They'll have like allele frequencies that are in between what you find in the UK or whatever in Ireland and what you find in Scandinavia. So that was the kind of the main point there. Okay, let's move on, I suppose. So what does DNA data look like? This is just sort of a like just like how is it, like, we talk about A's and T's, but like what does it kind of look like actually on the computer, or like what do I kind of think of it as in some way? I don't know, that's not the right question, but I don't know. This is like how it's stored at least. So basically, we need a way of classifying alleles at every SNP. So the classic way of doing this is it doesn't really matter. You can actually flip it to be 210 from what I say, or you can have some other way of saying something like do like minor allele frequency, major allele frequency. But generally, the simplest thing, in my opinion, is to and the important thing is to keep it consistent. Is you can do like 0 for ancestral, 1 for heterozygous, and 2 for derived. So basically, what it really is saying is the account of derived alleles. So if you have both the ancestral copies, you have no derived alleles. If you have one derived copy and one ancestral copy, then you have one derived allele. And if you have two derived copies, then of course you have two derived alleles. And then because due to your sequencing error, structural variation, or like you might just actually be missing a bit of your DNA, or someone that might have some extra or whatever, or just have low coverage, which is very much a thing of ancient DNA, you will often have missingness. So you need to have a way of representing that, which we just do 9 generally, but it doesn't really matter. Anyway, this is just sort of a simple way of keeping track of things, essentially. And so we need to kind of compress that data into like a small but easily readable form. And a very simple way of doing this is to do what's like called a matrix, which people who are familiar with math and whatnot might be familiar. It's not the movie, but if you're not, but it's just, I don't know, it's in the term. It's maybe more jargon, but oh well. And it's just sort of like a table. You can kind of think of it of numbers, and I'll kind of show you on the next slide. And it can be transposed, means like flipped depending on needs. So let's go over to that next slide. This is the matrix. As you can see, I literally just a table the way I've made it, where you have n SNPs and m individuals. And I have it as the individuals being columns and the SNPs being rows. But like I said, you can transpose it, which just means like flipping it so that now the SNPs are the columns and the individuals are the rows. That's not the important thing. And so you can see that, like, we have for each individual, you kind of go down the column and look at their genomes. The first SNP, they have a 2, so they have 2 derived, then 2 derived for individual 1, and then 0, and then 0. And they have a missing thing at SNP 5 for whatever reason, and then SNP 6, they then have they're heterozygous, and you keep going down. I made up all these numbers, they're not real, but just looking at them, you can kind of see maybe some people who are more or less related to one another. It's actually been a while since I made this slide, but I did intentionally make it where like I kind of made a couple people be more similar to one another. So like, I don't know, you can see like individual 6 and individual 1 are kind of quite similar, but like compared to maybe individual like 4 or something, right? So you can already kind of just visually see that there are like some of these differences. Of course, yeah, the human variation is quite subtle, but like, anyway, this is sort of just like a what it might look like. Yeah, so you have M individuals and then N SNPs, and you can do all sorts of things with this now. And so if you're curious, this is usually how like the data is actually stored in some sort of matrix like this. Okay, so this is sort of another problem, and I do want people to kind of think about this one. But like, which of these two scenarios gives us more information? So we have three populations which we know are closer related to one another than they are to the outgroup, which is the one on the side. And we want to know kind of just some information about the internal relationship of the some internal relation the internal relationships of the three that we have. And so which of these scenarios gives us more information, which are these two scenarios. So scenario one, we have one population that has a derived allele and then two with the ancestral state. And then the other one is just the opposite. We have two that are derived and one of the ancestral state. Which one gives us more information? And again, people in the chat can say, or you can say Jackson, or I can just like go into it either. I don't know. Again, this is very class-like, but again, it's kind of derived from
No, that's great. Let's see if we can get input from the chat.
I'll spill on YouTube. Again, fun time to think about things.
Yeah, I'll give it a few seconds because I'm sure that my answer is an ignorant one.
No, 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 no. It's fine. And
Okay.
this is not like I'm trying to build intuition because like this is not something that's inherently intuitive. So I'm trying to build like some basic intuition on how to use this.
Okay. So my naive answer is these give equal amounts of information.
That is like the naive and like seemingly like it's like two of them are related and one of them's less related. So that is like the thing that almost everyone says. Like people in my class also the same thing. This is also what I would say if I like kind of hadn't thought about this more. And this is like it does look that way. That's the way it looks. But it is like a little bit of a trick because we actually the two derived ones give us more information than the one that has one derived. And the reason being is the whole double mutation thing. And so I should have drawn a little tree, but you can basically draw a little tree of this thing. I'll kind of maybe even go on to the next slide about like gene trees. I probably should have included it, but oh well. Anyway, well, we're not going to go on yet. Actually, I'll stay here. But the point is, if you can think of like these three populations, if there's a single mutation, it could have happened on the very, very end of the branch. It could have happened like it could have happened a generation ago. It could have happened just like immediately before you took the sample, right? And so this mutation that you see in the first example, where on like the D person, even though it's maybe there is some increased probability of the two A groups being closer related, in theory, you could have the D group be closer related to either of the two A groups and not actually like the one that is derived could be closer related to the middle one than it is to the one on the left, or the one on the left than it is to the middle one, because the mutation could have happened after the two groups diverged. So like a kind of in linguistics terms, like there could be like a sound change in the one that's derived, but it's actually closer related. If you kind of ignore that sound change, it's actually closer related to like one of the other groups.
Okay.
And
Okay. Yeah. I think I follow you there.
yeah. And now the
Like
other one, oh, sorry.
so German undergoes the High German sound shift, which makes its consonant set look really different from other Germanic languages. But ignoring that, a lot of other structural features show this is a Germanic language. It's still closely related to English, German, to English, Scandinavian, Dutch, etc.
Yeah, exactly, exactly. And I actually think you kind of inspired me. Another example maybe is like you can correct me. I may be miss saying things here. Like Grimm's law, right? There's a big change in Germanic, right? It's weird, but like you can actually see that it's like so even though like let's say like Hittite and Tocharian or whatever don't have this really intense sound change that Germanic has, and so maybe they have like consonants that are more similar to you know like I don't know Slavic or whatever, but Germanic actually is clearly due to like other reasons is actually clearly a a clade. It's like related to the other Indo-European languages to the exclusion of Tocharian and Hittite. Even though it has this weird change at this location, but like overall on the whole, overall it's like it's clearly that just happened to be a thing unique to it and not the other branches.
Yeah, and I think part of what you see in linguistics sometimes is just it's the very regularity of correspondence is what is a clue to relationship. So even though language A has, a P where language B has an F, the fact that everywhere there's a P and A is an F and B shows you that they're related because it's not a coincidence. I don't know what the exact analogy to that would be in biology. I mean, maybe it would be, this lizard has orange spots where this lizard has green spots, but all the spots are in the same place. So the regularity of correspondence between the locations still shows you that they have something to do with each other.
Yeah, I don't know. It's an interesting question with the if what slash if there's an analogy there. I think it's it's not. I'm not thinking something off the top of my head. I think that basically doing this sort of thing stuff with DNA, there's kind of an advantage where it's like it's very clear like what a SNP is. Like it's like okay, we have like it's like it's hard to, I guess, it's hard to quantize language changes. I feel maybe I'm wrong. In the same way, like it's hard to like make this like matrix that I made here. You can't like make this matrix for languages. And if you do, you have to like kind of pick things out. You have to like you have to just choose. You just say, oh, like look, like I'm choosing this word to be in my matrix and this one not to be. Either kind of sampled it, or I'm choosing like morphology or phonology, or you have to choose what these things are. At least like the SNP like area. Well, or for yeah DNA that you sort of we have a bit of an advantage where we have that kind of done for us. Where there's like a quantized like you get a mutation, like it hits a locus, it's like boom, it's a single thing. So I don't know.
Yes.
I think so,
I think
sorry.
I think you're right that it's much that the 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 setting up of the parameters to make those comparisons is more subjective in linguistics, unless you drink 5 Red Bulls and then read something that Chomsky wrote in the right mood.
Fair,
Um,
fair.
so
I haven't done that. I haven't had my linguistic third eye open. So but yeah, I guess I was thinking about it as I was talking a little bit. I think that honestly, a rare correspondence, it's like a single mutation. It's at a single site because unlike linguistics, where you have a sound change of like F to B or B to F or whatever the example was before, that would then like hit the whole language sort of. While in genetics, like if you have a mutation at a site, like generally it just like it's that site, and so it's nice and kind of quantized in that way. Um, so also there's a chat. That would make for a very interesting thesis. I don't know what Lasagna is referring to. Sorry. Um,
maybe
uh,
the quantizing of
yeah,
linguistic data.
I definitely think it would be incredible, and I think people are kind of trying. Like I think like the Hegarty paper, which I think we kind of discussed last time, is very controversial, and like but my understanding is like they were kind of trying to do that with the linguistic side as well or whatever. Um, so okay, that was what Lasagna was talking about. Okay, anyway, I want to quickly come back to this before moving on, which is we have the two derived, those two people, because again, we don't have double mutations, or those two groups doesn't really matter, but like, these two samples, they must be more closely related to each other than they are to the third one, because there's only a single mutation that causes that. And, it must have happened before the common ancestor of the two derived ones, but after the common ancestor of all three of those. And so that's why that one gives a little bit more information. Um, okay, so I kind of continue on that just a little bit. It's a slight tangent because it's not really super important for F4 statistics, but I think it's good to think about, which is the genealogy of genes. So you have a bunch of genes that are lying around in like gene pool or in like multiple species even. There's some sort of genealogy, there's some sort of family tree. And if you look at like a single SNP, not like or other non-recombining set of DNA, so we're not looking at the whole genome, which point there's like lots of mixture and you can have genes from your mom and genes from your dad, and so it's kind of complexities there. But at a single SNP, that single SNP only has a single parental thing. Like the copy from your mom only comes from your mom, and it either came from her mom or her dad. And then let's say it came from her dad, that then in him went what either was from his dad or his mom, right? And so like there's a single path you can trace essentially. Um, and so it's like there's like a true family tree that like is like a true tree. It's generally unknowable because there's often multiple plausible trees for like a set of data. You can make multiple possible ones. And because we have no admixture, it's a strict tree. And it also importantly does not need to follow the most common overall tree. So an example I have is, it's well known that Europeans have a bit of, and East Asians and Native Americans and any group that's not like from Sub-Saharan Africa, has some amount of Neanderthal ancestry. Um, and so there are loci where Europeans and Neanderthals are more closely related to one another, and they have a more recent common ancestor in this, like, at least these SNPs have a more recent common ancestor, than either of them do with Africans. So it's like something they're more closely related with each other to the exclusion of Africans. Of course, they are also very closely related to Africans, but like more deeply in time, essentially. And so even though Europeans overall are much more closely related to Africans, and we're both like Homo sapiens or whatever, and so yeah, basically the the Europeans can have sites where they don't follow the overall tree. And kind of more interestingly, because this follows the kind of the admixture tree, but actually because like you have variation that just kind of hangs around, there are also, although much fewer of them and much more rare, um, there also are sites where actually Africans are more closely related to Neanderthals to the exclusion of Europeans. Again, this is kind of the least common site, like the least common pattern, but you do find sites where that is the case, basically. Um, Anyway, so admixture is one of the ways that these sort of discordant trees, or like trees that don't follow the overall tree, which again, the overall tree is like, you have Neanderthals here, and then you have humans there, and then within humans, there's like internal diversity or whatever. Um, but so you can have a discordant trees where actually, some group of humans is closer to Neanderthal than is to other humans. And this can be because of admixture, but it also can be caused by incomplete lineage sorting, which is where there's like polymorphism, or like there's variation basically in the ancestral group. Then they split off, and then like there's still variation in the like, daughter groups, and then they kind of randomly flip to being, they randomly, this group loses type A and this group loses type B or whatever, and it just happens that like some of these daughter groups, which are on like one side, end up looking like they're on the other side, even though they're not. But anyway, the incomplete lineage sorting is a kind of an important caveat there. Is it happens randomly, like so it happens all the time. Like actually, like I think 15% of the human genome is closer to a gorilla than it is to a chimpanzee. But also 15% of the chimpanzee genome is closer to a gorilla than it is to a human. There's this incomplete lineage sorting happening, but it's like even. So like 30% of the genome overall is in incomplete lineage sorting. So like 15% of the human genome is closer to a gorilla than it is to a chimp, but another 15% is equally related to both. It's an outgroup to both. And basically like when you have this sort of kind of random, it's basically the way you can think of incomplete lineage sorting is you have like both everyone gets like both copies, and then everyone randomly chooses which one they want to choose. And so like, chimps happen to choose one that's the opposite that we chose, basically. But they're going to have that as much as you have chimps choose the same one as gorillas, is going to have the same amount as everything, basically. And so that sort of it like doesn't yeah. So but well, if you have admixture, if you let's say you had like, actually let's use the real example. If you have Neanderthal genomes that enter Europeans, versus entering Africans, even though there are discordant trees where Africans and Neanderthals share to the exclusion of Europeans, there's more of the European Neanderthal sharing because of this admixture. That was kind of a lot. I think I maybe said it in a roundabout and confusing way, but hopefully that makes sense.
No, I think I get what you're saying. And it's, again, trying to draw language analogies, which aren't always perfect. There are going to be places where you take three languages related to some degree, A, B, and C, where A and B are largely more similar to each other than either one is to C, but occasionally A and C will pattern together and B and C will pattern together because of some random retention that's in common or some random innovation that's in common.
Yeah, I think that's exactly it, basically. I think that's a good way of putting it. I mean, there is like, of course, it's never one to one, because you do have like this like kind of forced shared like variation in the ancestor, but I think that in the broad strokes, I think that's a great analogy. Good to kind of rehash everything, even though it's the same day. So basically, an allele frequency is the rate between 0 and 100%, at which an allele appears in the population, and generally this is the derived allele. Although again, it doesn't matter as long as we're consistent. So let's define some things. Given a population A, the allele frequency at SNP I, which I is just like a numerator, right? So you can have like, 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? It's just some arbitrary number I. At SNP I is A sub I. Similar to the population B, we'll define the frequency to be BI at this SNP I. And this holds for any other population. So if we had population, I don't know, ZI would be the allele frequency. This is just to have numbers that like that way I'm putting things down, people know what I'm talking about. And the general allele frequency of A is like A. So if I'm not talking about any specific SNP, if I'm just like kind of talking more theoretical terms, then you can just kind of consider the allele frequency to be like lowercase a. Um, and yeah, basically, Um, yeah, I kind of say when I'll be using that A, like if I'm using like expected value of the SNP or if cases we're not concerned about a specific SNP, but that just sort of yeah, and like kind of we're talking in theory land, we'll sometimes use that. And then this also applies for other populations. So, for B, again, lowercase b is the general allele frequency. So these are just terminologies, which I want to make sure people know. So when I'm putting these things down, people aren't confused what I mean. But if people are confused, please just let me know. Okay. So what is treeness? So we kind of talked about trees a little bit, but we were going to do a test for treeness in a second. And before we can do a test for treeness, we need to know what the heck we mean when we say treeness. So we're going to use the graph theory's definition of a tree. So people are familiar with graph theory, which is kind of like a branch of mathematics, will be familiar with this, but if you're not, that's totally fine. I don't expect anyone to be. So it means that there's no cycles in what's called a graph, where a graph is sort of a set of nodes. Here I've drawn them by circles, connected by edges. Um, and basically it means if you kind of go along a path, there's no way of getting back to a node without kind of turning around almost, reversing course. So like I don't know if people can see my mouse. In the first single one, it's a tree. It doesn't really make any sense, but like you can't. I mean, maybe it doesn't even make sense to ask, but I would call it a tree. It's like a singular node. The second one, we can get here, but to get back, we would have to reverse course. So again, it's a tree. There's no cycle here. And the third one, again, we have now three nodes and two edges, excuse me. And again, we can go here and go here, and we can not never get back unless unless we reverse course, so it's a tree. But if we add an extra edge, then we can then make a little loop. And so this is now what's called a cycle. And so it's no longer a it's no longer a tree. And that's you can see that we have some more examples with four nodes instead of just with four nodes instead of three. And I want to point out here, I suppose, that like you can have like this guy here, this kind of box looking one has four edges and um is not a is not a tree, but then this one here actually does have one less. But you could make this a cycle actually by adding you could remove one of these and move this like here, and you could have basically the amount of number of edges don't matter as much. I should have thought about that before I said it out loud. I was kind of thinking in my head, but the point is you can have like different types of cycles, and it doesn't need to be like a one big circle. You can have this one, which is kind of more of a weird shape, and even if you didn't have this edge here, you just have one cycle, and then this part is not part of a cycle. It still would not be a tree. To be a tree, the whole thing needs to be tree-like. That's I guess the thing I was trying to say, but was kind of thinking out loud. Anyway, so that's the ones that have check marks are all trees, and the ones without check marks or the ones with X's are not trees. So hopefully that's not too complicated. It's not super important, but I am going to use the word treeness. So what do I mean when I say treeness? Is it basically I'm asking, is it a tree? And for admixture graphs, which are like sort of kind of the family tree of populations, so you've probably seen these somewhere if you've ever read an ancient DNA paper, but like you can have like a picture where you have sort of like the ancestor of all hominins or whatever, and then you have like it splits off into like all the different apes or whatever, and then human ancestors, and then human ancestors you have like Neanderthals and then like modern humans, and there's like a branch that goes from Neanderthals into the modern humans, which is this Neanderthal admixture I kind of talked about before. So a tree is one of no admixture events. So this Neanderthal event would make it not a tree, right? Because then you'd have like a cycle, you'd have like some sort of loop. But if you don't have admixture, you would be a tree, and there's plenty of branching events. You can have like as many branching events as you want. And so, I think this is probably very familiar to people who have like, linguistic family trees, basically. It's the same kind of idea. I don't know if I've ever seen like a linguistic like admixture graph. I'm sure you can make one of like the borrowings that happen, but yeah, I feel like that's not quite as common. But you definitely see these like trees people make of different like languages. So it's the exact same idea, basically. Okay, I'm
Yeah.
gonna move on. Sorry.
Yeah, I think I get you. The visual really helps. I mean, it can't form a circuit. It has to be one directional, although, well, between a given node, you can't have two direct. Yeah, I get you.
Yeah.
The visual is what makes it make sense.
I'm glad. Yeah, there probably is a formalized graph theory definition, but if I ever do it, I definitely don't know it now. And so yeah, I'm glad with the visuals as well. And I'm glad that they're helpful. But okay, yeah. So how we move? I moved you the picture I have of you because in the way of something I want to see on the screen. Anyway, so also there's a comment on the chat. Um, It has a trivial. Oh, so Blake says it has a trivial fundamental group, if and only if it's a tree. Okay, so that's the graph theory definition, apparently. Also, thank you, Kate. I just saw it now. Okay, yeah. So let's talk about the F4. So the F4 statistic is probably the most common statistic you'll see, as long as the F2 and F3. Um, okay, so there's the apologist way of looking at it for the previous thing that Blake said, apparently. Anyway, so basically we want to test if these we want to test if the populations A, B, C, and D are a tree. If they are a tree, specifically the tree like this. We're not testing all trees even. We're testing for treeness with this specific tree. So like the way it's written out, you have F4, like population 1, comma, population 2, semicolon, population 3, population 4, right? And so F4 A, B, C, D test if A and B are equally related and C and D are kind of equally related. Well, A, D, B, C test the second tree I wrote, where it's like, are A and D equally related and B and C equally related. And for simplicity, for now, we're just going to, this is not necessarily the truth in like actually computing them, but we're going to assume we have a single haploid sample. So we have a single individual that either has a P or a Q, right? So it's either ancestral derived. And so in case of the frequencies is neither 0 or 1, the same logic will be a thing, right? Like if you can actually have like values like 0.3 and 0.4, and we'll come back to that, but it's not, this is not, yeah, don't worry about it so much, basically. This is the long and short of it. Anyway, basically, yeah, so we're testing these two trees, and I should point out though that if you compute the test for tree 1 on tree 2, it might reject it actually, because they'll say, oh, look, like B and C are really closely related, and A and D are really closely related. So this first tree can't be a thing. There has to be some admixture between B and C and also between A and D, right? If you had if you had the second tree be the reality and you did the first test for A, B, C, D, it would reject treeness, even though in fact it could be a real tree. So that's like a maybe subtle thing, but like it's not just a test for treeness, it's a test for this specific tree that I wrote out. So again, the first F4 A, B, C, D test the first one, and A, D, B, C test the second one. But it's not a general like but I guess the important thing is the order matters. The order of the 4 populations matters. So it's not a general test for treeness, it's testing a specific tree, basically. Blake is giving another definition of trees, which is interesting. I don't know if I'm going to read it though, just because it's a little bit long, and we're I guess kind of moving on from it, but it is interesting. Um, anyway, does that make sense on the like what an F4 is testing, by the way, real quick?
Yes, I think I
Okay.
understand you.
Perfect. Okay, so real quick, I'm gonna talk about like a rooted tree, and this is all important. This is all a bunch of setup, but I think it is important for like understanding kind of the logic and intuition. So a tree is rooted if we know what the root is. So I again give little check marks where it's sort of like it basically means there's like sort of an arrow of time, and we kind of know where the first split is. And so we like we have some sort of route on the first floor, we can kind of see the directions, but on the other one it's sort of we have like we don't know which one of these, like on like the bottom left, we don't know which one of these like 3 or which one of these 4 like kind of groups that we have come off from the middle is like the ancestral one, right? So like clearly one group is splitting off and then 2 other groups are splitting off, but like the ordering of like that kind of depends on which one you choose to be the root. And it's so we sort of don't have information there, basically. Like it's sort of an unknown thing. And that's sort of it's interesting to point out basically that like in the way you can think about it is if you didn't know the order of time, if you didn't know like the direction of time, you could say that like this kind of sound kind of weird, but like you could define like vertebrates to be a thing, and you could say like vertebrates are like these groups to the exclusion of everything else. But if you didn't understand the way like evolution went, like the direction of time, you could define like invertebrates rather than being like all animals without a backbone. You could define it as like everything that's not a vertebrate, including like bacteria and plants. And sort of maybe like if you get rid of the root, you get rid of like direction of time or whatever. You can say, oh, anything that's like not part of this group is like a different group of its own sort. It's a little bit weird, but anyway, it's like it's just important to think that that like we can kind of do this in if we kind of ignore time, we can sort of we can sort of we can make these unrooted trees. And for people who are more mathy, like Blake perhaps, the like process of allele frequencies changing over time is what's called martingale, which means the expected value is the same between each generation. So like, if let's say 50% of the people have a T and 50% have a G, the next generation that should also be true. Of course, it could change if like just randomly happens that the people with T's have more kids than the people with G's. But unless there's selection, the expected value is the same every single time. And there'll be some noise or whatever to it because you're never going to get exactly, if you do a bunch of coin flips, you're never going to get exactly half heads and half tails. But like in theory, it should stay where it is at the last generation. So each generation will update based on like the generation n plus one, the expected value is generation n. And that also goes backwards in time. So anyway, the basic drift doesn't really care so much about time. So we can make an unrooted tree. So hopefully this will now put everything together. So we can manipulate these trees in like an unroot. We can manipulate unrooted trees because we don't know which one's the actual ancestor. And so F4 statistics are testing the treeness of an unrooted tree. And so we can and they also don't care about branch length for what it's worth. So like a really long branch, it's the same thing as at least the terminal branch length, like the branch between like A and the common ancestor of everything else or whatever. And because of that, we can kind of stretch the branches and kind of reshape things. So this tree we had before, this ABCD tree, we can make it out of this little like sideways thing. You can see I have like I'll use my mouse again. So we can take this and we can just turn it into this tree. We can go boop, and we can kind of like take a little break right here and kind of twist it downwards. And we now have like this A, B, C, D tree. But we also can just sort of like stretch out this like branch. We can just stretch it out right here. And then we can sort of make a kink here and then like just kind of turn this downwards. And then like, we now transform this. Like this branch is now like this branch, including the kink. This branch is like still here. And we've just kind of turned this little like, I don't know, not triangle, but this little like tripartition thing, this little like tri-piece or whatever. And we kind of turn it down and now it's this piece. And so these two trees in F statistic kind of fashion are like equivalent. Obviously, they're not. Obviously, they're quite different from like what actually is happening like genealogy wise. But if you were to compute an F4 on both of these trees, they would both be like, assuming this is actually the true tree, and we were to compute F4 A, B, C, D, it would be 0. It would be like, this is a tree. It can't reject treeness. So does that make sense? That's why we went to this whole unrooted thing, which may be a little bit odd. But it's because that this is an important thing to understand that an F4 statistic, it's checking this tree that I have here, this unrooted tree, right? It doesn't actually care about the ancestral state. And so like A, B, C, D, where A is like a close relative of B, is the same thing as A, B, C, D, where A is an outgroup to everything. The important thing is that A is equally related to B from everything else. Like if you were to traverse from A to C or D, you're first going to pass by the area where B gets to traverse to C or D. Like A is equally related to C or D. And it's the same thing for B. If you're B, you go from B to C or D, you have to hit this location, which A also has to hit. You basically you have this like shared branch. This branch is totally shared between A and B and when you're on your way to C and D, basically. And the same is true for C and D. If you're going from C to A, you have to hit this point first, and you have to like go through this like shared point. And that's like the same no matter how you kind of stretch out the branches. Does that make sense?
Yeah, I think I get you.
Okay. If people in the chat again are ever confused, please just say it. Okay. So let's talk about ABBAs and BABAs. Oh, I messed up the capitalization. That's unfortunate, but oh well. So ABBAs and BABAs are just a fun little way of remembering sort of what are the basics of like an F4 R, which is basically we count the number of times we see 1 0 0. Oh, I'll come back to it. I'm going to read out my text first. I think it'll build it easier. So okay, we want to find the F4 UVXY. And as a reminder, that is testing the treatise of UV and XY. So are U and V kind of a clade in some way compared to X and Y? Again, in an unrooted fashion. So U could actually be an outgroup or V could be an outgroup, as long as the U and V are equally related to like when you compare them to X and Y. So given our sample at every SNP, each population either has a frequency of 0 or 1. Again, we're assuming we have a single haploid sample. And so we can make a table of the first 9 of 16 possible observations, because there's 2 to the 4 observations. And actually, there's really only 8 observations if you think about it, because the 0 0 0 0 is the exact same thing as 1 1 1 1 1, right? Like if you flip the especially we have an unrooted tree where we no longer know the where the ancestral version is. I know I talked before about like how you can have actually differences of knowing ancestral and derived, but like in this like time scale, basically, we can say like, we don't really know anything. Um, yeah, basically the if we flip the ancestral and derived, actually, yeah, if we flip the ancestral derived, it's like we basically don't get any more information. So like the 0, 0, 0, 0 is the same as 1, 1, 1, 1. So of these 16, which of them are useful for answering questions about treeness? So obviously the first one's not. The first one, if you have a SNP that looks like 0, 0, 0, 0, we've gained nothing. We've like, it's okay, everyone's related equally. So who cares? And if we have this 1, 0, 0, 0, we also gain nothing. This is kind of more like the previous example, which was to build intuition. Because again, like, we know that U is different from everybody else, but it doesn't tell us who U's closest relative is. Like it doesn't tell, you could have just had a mutation or could have a really long branch, which again, we set up our statistic actually purposefully to kind of ignore branch links. I kind of mentioned it here, but like, a really long branch, the same thing as a short branch. So this one right here, it doesn't do anything for us. And that's the same thing as the one that's just V, and the same that's just X, and the same just Y. And so then if you want to answer a question about treeness, the one that is like like 1, 1, 0, 0, that might seem interesting and important because it's an example of treeness, but we're trying to reject treeness, not like prove it in a way. We're trying to basically what F4 is actually doing is it's rejecting treeness. And so we can't reject tree, like it's evidence in favor of treeness. We also ignore that one. It's a little bit confusing. But the one that like goes 1, 1, 0, 0, and then correspondingly 0, 0, 1, 1, we actually we're gonna ignore that one basically. But on the other hand, the one that where we have like 1, 0, 1, 0, where we have U and X like right here being closer to each other than V and Y, this is evidence against the tree. This shouldn't be happening basically. And if it does happen, basically, it should happen as much because maybe it's incomplete lineage sorting. That's a possible thing if they're like these are recently splitting populations. And if that happens, it should happen as much as the opposite one, the 0, 1, 1, 0, this sort of V and X being related. So if you have it where you have a weird relationship between U and X, you should have an equal amount of relationship between V and X. And the same thing is true the other way around, where if you have, V and Y, you should have equal amount of, like X and sorry, if you have V and Y, you should have an equal amount of a U and Y, right? So you should have like an equal amount of these sorts of ABBAs, which you can see ABBA is this 0, 1, 1, 0 or 1, 0, 0, 1. And then BABAs are 1, 0, 1, 0 or 0, 1, 0, 1. They're actually the same. Like you can see that we're just A or B basically. We don't actually care about which one's ancestral derived in this case. It's just to keep track of things. So that's like what we're looking at. It's the number of ABBAs versus the number of BABAs. And assuming the tree is correct, maybe move on now. Yeah, so I kind of say we can flip the zeros and ones. So we can just kind of simplify this to we have AAA, BAA, and when everyone's the same, it doesn't, we don't care. And when it's one, it doesn't tell us anything. I kind of already said all this. I probably should have flipped the slide earlier, but oh well. Anyway, basically, the only the ABBAs and BABAs give us information to reject treeness. So you do expect there to be some of them, again, due to incomplete lineage sorting, but the numbers should be relatively even. And so an excess of either ABBAs or BABAs, but not both, like they have like an equal amount, you don't care, then rejects treeness. And you can, again, like using like a standard error, you can then get like a z-score and find a p-value and sort of reject the treeness very strongly. And there's also an interesting thing. So the D statistic, I give some math about this. Don't worry about it. But it's like the actual like what the D statistic is, just like kind of a special case of the F4 statistic, which is like scaled in a nice way. Basically, is it's the number of BABAs minus number of ABBAs divided by the number of BABAs plus ABBAs. And that's like what the value of the D statistic is. And so yeah, basically it gives us a non-zero result, rejects treeness. And what's interesting is it also a significant value will also tell you the direction in which the tree is rejected. So if you have way more BABAs than you have ABBAs, for example, you have way more, so if you have a positive value, that means there's some relationship between either the first and third population, the second and fourth population, or both. It actually doesn't tell you which case it is, but like, perhaps there's admixture between U and X, or X into U, or V into Y, or Y into V, right? It doesn't give us the whole story. Like we don't know everything from this, but we then know that there is some sort of relationship happening. And you can actually make this simpler because let's say you make U be an outgroup, you make U be some group that's like, outgroup to everything else, then you know that it can't be a U and X relationship. So it must be a relationship between V and Y, basically. And so that's like that's sort of a positive D statistic. And then if you have a negative one, it just means the opposite. It means that there's a relationship between V and X, right? Or again, U and Y, unless you have U or Y denoted as like an outgroup or anyway. Yeah, using an outgroup for one of these four can simplify things because you can see there's still a lot of options once you reject the tree. You can reject the tree, you can then even know with like strong p-value which direction the tree is rejected, but then there's like four options basically therein. So anyway, that's what an F4 statistic is. And so you can do things now like already with it. And let's think of it like applying it for like Indo-Europeans, which we kind of talked about last time, right? You can like you know um say that like if you take, yeah, I don't know, like Indo-European group like one, Indo-European group two, like or like outgroup Indo-European group one, Indo-European group two, and like non-Indo-European group, then like you might expect if they like have shared ancestry, these two would be like related, and thus you get like a negative F4 statistic. So I mean, or a negative D statistic. But again, the D statistic is really just a special case of the F4, where it's sort of normalized and scaled by the Abbas and Babas. So anyway, yeah, hopefully that all makes sense.
I think so.
Okay. So now let's talk about F2s, which I want to talk about F4s because I think they're a little bit simpler actually. This like Abba Baba thing is really clean and it's quite easy to see like visually. But now I want to talk about what an F2 is, which is kind of a measure of drift. And then we'll kind of go and build up actually back up to the F4 statistic from the F2s. So an F2 is a measure of drift, like I said, it's a measure of difference basically. And so it's denoted as F2AB with like a semicolon between it, and it's equal to a minus b squared, which more formally, kind of a longer definition, is for all SNPs I, like for sum over all every SNP I. You take so essentially it's an average because you divide by the number of SNPs. You take the AI minus BI times AI minus BI. So it's again AI minus BI squared basically. So it's the squared average difference in allele frequency between two populations. That's like a English definition or whatever of the saying the exact same thing I just said. And it's a measure of total average difference between two groups. It'll always be positive. Hopefully it's clear as to why that is. It's because it's squared. So, it's even if these are, even if, you get a negative value for A minus B, it'll be positive. You have the opposite will also be positive. And we can kind of make this little graph here, which is not super useful for F2s, but will be more useful kind of as time goes on. And we keep building up our intuitions on these different things where if you have a population A and a population B that both descend from sort of ancestral population, which also known as R for root, the more time that's gone on both of these like branches, the more separation there'll be. And you can, again, we think of allele frequencies as being martingale. So like every generation, they're expected to be like about the same. Like, if you should change at all, but like it kind of randomly kind of wiggle. And so, randomly wiggles this way and then that way. And like, there's changes, but like there's no rhyme or reason for why these are happening, right? It's just sort of a random path, it's a random walk or whatever. And so, they both start off at the allele frequency of R. They both start off at RI, this root population, this ancestral population, and they kind of meander along. And they might even flip back and forth. They might not. They're not always going to go in the same direction. As they get further, the distance between AI and BI is going to get larger and larger. So as time goes on, like A minus B squared is going to get larger and larger and larger until eventually it fixes. Eventually, infinite time, A will, end up in one of these two places and B will end up in one of the two of them. And it actually could end up at zero because it could be that like A and B are the same here and the same here. They could fix together, just happen to either both end up at zero or both end up at one, or they could end up at opposite, ends of the spectrum, at which point there would be the maximum F2 at this locus, which is one. So anyway, and so also because of that, by the way, the F2 is bounded between 0 and 1. So you'll never get an F2 larger than 1, basically. And if you have an F2 of 1, it would be kind of impossible because at every single locus, basically, you're like opposite, essentially. Every locus, I guess, that you care about is every SNP. So maybe it's like you're totally inbred, basically, is what it means if you have an F2 of 1 between two populations. So, okay, I'm gonna move on, but please, like I'm saying, like stop me at any time for anything that's confusing. Okay, let's talk about F3. So F3s give us, this is just sort of an interesting thing that can be used to look like differentiation and difference. And, it's like, it's interesting, it's useful. And there's some interesting theory about how you can kind of build F4s out of F2s, but that's not for today. But it doesn't really answer a question. So let's go back to answering questions. The F4 answered a question. Is it a tree? The F3 can be used to answer a question. Is it admixed? So basically, the F3, first we'll go to the definition and then we'll go on to this little drawing I've made. But basically the F3, if you have F3 ABC, and well, it's important that this semicolon is really important here because you could also draw like CAB or whatever, but like the semicolon really distinguishes the single population versus the double population. So you have F3AB semicolon C. So what this is it's I guess I'll do the simple one first. It's A minus C times B minus C, which kind of you'll see this is like these almost the same formula as before. It's very similar, and all these F statistics use a very similar formula. This is just A minus C or AI minus CI times BI minus CI for all SNPs kind of averaged over. And so there are multiple uses of the F3, but the most basic is to test for admixture. And so why would a negative F3 imply that C is admixed? Also, I should note before I even go into this that a positive F3 does not guarantee that C is unadmixed. And I'll come back to that, but that's an important caveat. A positive C does not reject admixture, but a strongly negative F3 strongly implies admixture. So why is that the case? Why is the case that this will show admixture? So, we have our little like phylogeny of different populations. So this ancestral population that creates this ancestral A, and then ancestral B, who then mix together from ancestral C. And then there's more time that goes on, and we get our modern A, our modern C, and our modern B. And there's no mixture after this point. Just it's a very simple little model. So basically, what happens is you can see on here if C is an admixed group of A and B. In this point, I made like a 50-50 pretty even mix, but it could be like 3/4, 1/4. It doesn't really matter. You'll end up being C will end up being somewhere between A and B, right? If you form C out of a mixture of A and B, it inherently will be between A and B when time starts. So A and B are starting off kind of similar to last time, kind of randomly moving. I drew this, so it's probably not truly random. Humans are pretty bad at making things that are truly random, but in theory, think of it as kind of randomly moving apart. There's no rhyme or reason to it. It drifts because of randomness will always kind of drift, but like it's just kind of moving on its own. And then there's a certain amount of differentiation at this time t or whatever time this dotted line happens, and we now form C. C immediately starts off as in between A and B. So A minus C, if it's positive, then B minus C will be negative. On the other hand, let's say it just so happened that B was above C and A was below C, then A minus C would be, in this case, negative. It's not, obviously. But then because we flip them, then B minus C would be positive. And so whatever value whatever like, I don't know, what's the, not cardinality. I'm forgetting the term, but like whether if one is positive, the other one is forced to be negative if C is in between them. And so, of course, over time, things continue to drift. And at this point, C is a lot less between. It's kind of more, it's more different. But in theory, if enough time had happened, C could escape. C could like end up over here, and A could end up down here, right? You could like right here they're pretty close even, right? So like in theory, but due to random movement, C could escape from these and then no longer be negative. And so if it's been a long time since admixture, or if the admixture was very subtle, like let's say there's a very small amount of A ancestry, so C starts off over here, right? Then it might end up really quickly escaping kind of the bound of B in some way. And so if C can escape, at which point it's no longer negative. But assuming that it hasn't yet like kind of escaped from its parent populations, then this orange line value, this like A minus C value, will be the opposite of like in direction of this B minus C value, which is green. So again, in this case, this one's positive and this one's negative. But if you flip it around, it would be the opposite. And so this can test for admixture. So again, kind of maybe trying to make a real world example of it. We could like look at populations in Europe and say, are they admixed between the farming populations that preceded them and the like Yamnaya steppe pastoralist people that then came in? And if you tested that, you set up like, C is modern European, A and B are like, again, like the steppe pastoralist and like a farmer, you would get a very strong negative signal. And similarly, you could also go to India and kind of find the preceding farmer groups or hunter-gatherer groups. Like it is not unfortunately as good of a source for like because we only have ancient data for like northern India, but we do have it. We do have it. But so you can have like Indus Valley or whatever, and then put in like the Yamnaya again and test it, and you'll again get and for like modern Indian, and you'll get a very strong signal of admixture. So you can use this to test things. And then you also could, in theory, put in like some other groups. Let's say, you wanted to test if it's like shared Iranian ancestry, let's say, because that's like the Hegarty model or whatever. You could put it in and put that in for um, B instead and see what it tells you. And it is possible that it would still be negative for both India and for Europe, because it's true that there's like this Caucasus ancestry, which is kind of related to Iranian ancestry, which is part of Yevnaya and does move into Europe. And so it's possible they would still show it would be negative, but it'll probably be less negative. It'll probably be less intensely negative than the Yevnaya ancestry. So, and it might even be like, especially for Northern Europeans, it might be positive. It may not be, it may, it may not be maybe so old, first of all, because it is an older admixture, and also so subtle because it's like less of the ancestry that it will have escaped. And so that's like maybe evidence you can use to say, well, it's probably the Yamnaya that are spreading it, especially if you assume it's an equal amount of admixture and kind of an equal time versus the like Caucasus ancestry. I'm kind of making this up on the fly. I don't know if this is all actually perfect, so I may be making a mistake here or there, but I'm trying to relate it to like specifically like Indo-European genetics, which I think maybe is where people are like where the crossover is between people who are interested in the stuff I'm interested in and the people who are interested in your channel. But yeah, anyway, so yeah, hopefully that all makes sense.
Yeah, I think I'm following.
All right, perfect. And I don't see any comments in the chat, so I'll keep going. But yeah, so that's the F3. Okay, so you also can use the F30. I mentioned before you can use it for it's mainly used for admixture, but you can also actually use it for to test for like outgroupness, to test if a group is an outgroup. And so I have a kind of a different setup here. We have some sort of ancestral population R, like prime or whatever, the ancestral R, which then forms like later R, which then splits into A and B, and also into like ancestral C, which turns into C, right? So you have some sort of like genealogy of these like different populations. Eventually with A, B, and C, where A and B are kind of a clade of one another, like a related to one another, the exclusion of C. Okay, so just like how C is admixed, like if C is admixed, F3 is negative, a true group will be like positive. It'll actually be strongly positive. And it'll be more positive the more distant C is, or the closer A and B are. So this basically is a test for a kind of outgroupness, but actually it's more interestingly can be used as a test for kind of sharing between A and B. And you can sort of think of it as kind of measuring this branch length. So it kind of measures in some way. This is like an advanced I have another lecture on this kind of advanced F statistics, but it measures this like branch. And so if this branch is larger, which either means C is more distant, right? If it's like further out, or if this part's more distant because A and B are closer, then it'll be a bigger value. So that's kind of the intuition there. So why is this? Basically, you have R prime, and that eventually goes to R and C prime, or whatever, and the allele frequencies shift over time, as so the same thing we've been doing before. And at a certain point, then R splits, and we now have A and B, and you can see that this value now of A minus C, B minus C, which is it's the same value we had before, because it's the same statistic, it's the F3 statistic. This value now is essentially what it's giving is it's giving like it'd be the same direction, and it's giving like how different C is from A and B. And if these things are closer together, then the like it's sort of like if you take like if you have a total of seven number, or it's like eight numbers to put together, like you've eight things, and you want to divide them into two piles and multiply those piles, which will be the largest pile. This is just, I'm just going to state this, and I'm going to give a little bit, but I'm not going to prove it. So if you more homework if you want it. But if you have like eight numbers or eight like units, and you can divide them into two piles, and you want to multiply those two piles to get the biggest number. If you do like 2 and 6, you'll get 12, but you do the best when you have the piles be even. So if you have 4 and 4, it'll be 16 as the largest. And that's kind of the intuition I would give. Versus like 1 and 7. If you have 1 and 7, then if you're two piles, then you're going to have 7, right? So like the best you can do is at the even distribution 4, 4, and that gives you like the largest value. So that's kind of the intuition I'm going to give. I don't know if that's one to one, but that's the way I kind of think about it in my head. But basically, if A and B are more similar to one another, the value will be larger. And again, if C is more distant, again, the value will be larger. And so this can be used to test for relatedness between or shared drift between two groups. So basically, if you have, if A and B are closely related, you'll have a larger value. And so this is kind of an interesting thing you can use to test for relatedness, basically. Anyway, that's the outgroup F3, which also is quite common in these sort of ancient DNA papers. Everyone still following, Jackson?
I think so.
Okay, yeah. And I also don't say anything in the chat, so I'm just gonna assume people in the chat are okay. Yeah, someone actually does say something in the chat. Yep, awesome. Okay. So let's return to F4s. This is kind of a more monstrous thing now. You have way more like compared to the F2, which is nice and simple. This is like getting quite intense. So which is part of the reason I introduced the F4 differently before. But let's kind of think of a different intuition for the F4. And I gave my Abba Baba thing, which is like a proper intuition. It's not wrong. It's not like a simplification. That's like that. The Abba Baba thing is how, for example, the D statistic is like based around. But we can kind of think about it also in this sort of like allele frequency over time graph, basically, that we had before. So we're going to use the same framework, and formally, you can see the F4 is just A minus B times C minus D now. And this, again, the semicolon is quite important here. You have A, B, semicolon, C, D, and it's the sum of all A minus B's times the sum of all C minus D's divided by the number of SNPs. Also, I see a couple of chats. Okay, you can prove the maximum thing by finding the vertex of x times m minus x and observing how this happens at m divided by 2. If anyone wants a hint of how to solve it, from Blake again, who's very clearly a math person. I think he mentioned that as well. Anyway, so yeah, so basically, yeah, so you have this definition. Again, it's very similar. You can see to the F2 and F3 definition. Oops. You can see I'm basically out of slides. I'm definitely going faster than I intended to. But it's similar to this F3 definition, and it's also similar to this F2 definition. It's the same thing. But this time we have 4 unique populations. So basically, we have A, B, C, and D. And the F4 statistic will be strongly positive if and only if the difference between A and B is correlated with the difference between C and D. So basically, if there's no correlation, if it's totally random, like in this chart, it is totally random, and so actually, like in theory, these will like be quite small or whatever. But like if this is random, the drift from like X to Y is random, but there'll probably be some difference because there's drift. So you assume there's some difference, but we don't really know how much there is. It's kind of random. And if we then have like we then are equally likely to have D above C or D below C, right? It's random drift, right? Assume there's no admixture. So it's random, and this is random. B is equally likely to be above A as is to be below A. It's just kind of it's random, right? And so overall, the average over all of the loci, if you or average over all the SNPs, excuse me, you assume that basically there'll be the equal amount of times when C is and B are both over A, C is over D and B is over A, as you have the opposite, basically. Like it'll always kind of even out. But it'll be positive. You get a positive value when there is some sort of correlation here. When like, let's say A is over B and C is over D, right? Or if it's flipped, if D is over C and B is over A, consistently. If it's consistently a thing you find, it'll be positive. Because basically what that means is consistently A minus B has the same sign as C minus D. And maybe I'll go into like why that would cause it with admixture in a second. But just first thing about that, it should make sense that this will be consistently positive if A minus B has the same sign as C minus D. It doesn't matter if it's positive or negative, but like it must have the same sign. On the other hand, if you have it being significantly negative, they must be kind of anti-correlated, right? Like whenever A minus B is positive, C minus D must be more likely to be negative, right? Like A minus B must be the opposite sign of C minus D consistently for this average to be kind of strongly negative. So I hope that all makes sense. Again, don't think about too much of to why. I'll get that to there in a second. But like the A minus B sign, if it's the same as C minus D consistently, it'll be positive. If it's the opposite of the C minus D sign, it'll be negative. And if it's just random, then when you average over the whole thing, it'll be it'll be zero. It'll be, it'll be statistically it'll just be noise essentially. If you have enough SNPs, it'll go away basically. The p-value will be very, you can't reject it basically. For like rejecting the treeness. So now let's think about like, okay, everyone follow on that first. I'm gonna wait for the chat also, but also Jackson, if you have any, if what I just said made sense or didn't.
Well, I appreciate you walking through the F2 and F3 before coming back to talk about this with F4, because it makes more sense built out of the smaller comparison sets.
Okay, Matt's good. Yeah, it definitely is a lot more. So yeah, I appreciate you saying that. So I guess that was my hope, was to kind of build it up slowly. But yeah, do you follow on the okay? I guess it sounds like the chat's pretty good. So I'll kind of, so yeah. So just reiterate before I move on. If the F4 is positive, that means that these two are the same sign. If it's negative, they're the opposite sign. And so the reason that would happen, for example, is like one reason that could happen is if there's admixture from A into C. So let's just keep this exact kind of randomness map, right? This exact randomness map we already have here. And we're going to now have C be pulled towards A, C be pulled towards A, right? So because there's an admixture from A into C. So we're going to just move C like down here. So now A, like it'll be closer to the direction that A already is. And essentially, when you have that, it'll be the basically it'll be stronger. Like that pull will be stronger when A is lower down, right? If it's being pulled down, if it's being pulled up, it's opposite. But basically, it'll be pulled more, even with the same amount of admixture, if A is further down than if it is like if like this was flipped. And so you're going to end up with C being pulled kind of towards the direction that A is. And if A is below B, you're even more likely, you're never guaranteed if it's a small amount of mixture, but you're more likely that C will just kind of go beneath D. It'll be pulled not to go beneath D. If A is above B, it'll still be pulled down, but it will be pulled down less. And so overall, you'll end up, I maybe should have drawn this out in like multiple drawings, but overall, what will end up happening is when A is below B, you will be like just biased towards, statistically more likely to have C also be below D. And when A is above B, it's still the same thing that happens, but it's less. You see, it's the strongest amount of it being pulled down is when A is below B. And if we flip this whole thing on its head, if we now have like say that X is above Y, X just happens to be above Y before this split off happens, we have the same admixture. Again, the pull is stronger when A is further up, right? And by the way, this is not like this. We're showing this as one SNP. This is like one locus, but there's actually like you have like millions of loci. You have millions of loci. So it's a small effect, but because you have millions of loci, when you average over all of them, you do get like a very strong answer, basically. So there is a lot of noise, and I want to be very clear about that. And that's also true for all of the previous things as well. These are all like singles, we're doing this on a single loci, and again, kind of averaging over the whole genome. Okay, so back to what I was saying. So let's say the ancestor of A and B start off above C and D, but there was admixture from A into C. So sort of you that have C and D down here, A and B up here, but C is moved upwards by A. It'll always move upwards, but it'll be moved upwards more when A is above D. So when A is above D, you are like extra likely to have C also be above D. And so you're likely to have A having like A's relationship to B will then be correlated with C's relationship to D, essentially. And I gave the AC example, but you can see this is true for basically all of them. And obviously, if we just flip who we're talking about, if we flip, instead of C, we talk about D, then the everything I just said is also obviously true, but we flip the sign because now it's now the CD thing is going to be flipped for the AB thing. This actually is simpler to think about. I maybe should have drawn where like A is an outgroup. If you have like A is an outgroup and then B and then C and then D, you don't have to. You can just obviously tell that the yeah, you don't have to think about so much the position of A and B relative to one another. But anyway, but if you want, we can draw these out on the whiteboard like we were doing last time and think about this in more detail. Because this is not the most intuitive way of thinking about it. Although I think it's in some way a little. I mean, I said it was not a simplification, but the Abbas and Babas, you kind of like when you think about them, you're kind of thinking about like a like A's and B's, like you're not thinking about them being like 0.35 or whatever, but actual allele frequencies are numbers between 0 and 1. So in some way, maybe this is more accurate. But I definitely think it's a little bit less intuitive. So, anyways, I hope I just gave a long spiel about that. I hope that made sense. If not, I'll try to go through it again. And we can also go to the whiteboard. I don't know how we're doing on time, but I assume I'm still going a little fast. Yeah, this is everything I had for today. Like I said, we can go through advanced F statistics if people really want to and are up for it. We can go to the whiteboard, and if people have questions before we do all those things, we can do that. And if we're out of time, we can also end.
Yeah, I'd be wary of going more than another 10, 15 minutes just because this is a lot to absorb
Okay,
and
yeah, no worries, no worries.
people get scared off from very long run times.
That's
Um,
fine. I mean, I'll stop share either way. But yeah, as you're saying.
I would say questions. Otherwise, I think the most useful thing to conclude with, well, actually the citations that you were talking about, but also
Okay.
maybe a couple whiteboard examples, like just like run through.
Yeah, I can do that. Let me do the citations first, just while I'm thinking about it, because otherwise I might forget, even if you remember Nicole et al.
I did not take statistics at any point. Uh, Sort of unbelievably, I probably should have. I think that has a lot of value to people who are in the humanities. Um, but like a lot of college students, when it came to getting my math credit, I was like, well, what's the easiest thing to do? Right? Um, and I took a class called contemporary math, which was just a blow off. And then I took logic. Logic helps to a certain extent with this.
Like
But
formal logic? Sorry.
yeah, formal logic.
I was a math, if people can tell, I was a math major for undergrad. So I'm coming at this from a very mathematical point of view. I actually also never took a specifically a statistics course, but having a math background, like taking like linear algebra and probability theory or whatever. Yeah, but I actually also somewhat regret not taking like a formal statistics course in undergrad. But I don't know, you can always learn on your own. I'm kind of doing that. Although again, I have an advantage of having a math background. But I think even if you don't have a math background, I think that there's so many resources online now, like MIT OpenCourseWare or whatever. So I think it's never too late, basically, for anyone who's interested and excited about it, for whatever reason. But I agree with you. I think statistics are, yeah, I mean, I'm biased. I like math basically, but I think they're a good thing to learn for any sort of, because you can, like you said, like I think people in the humanities often undervalue them, but you can always find a way of using them to test something, and kind of a way you maybe didn't even think about it at first. Yes, 2024. So I think I put the first one in already. Um, no, I didn't. I didn't enter. So here's the first one. This is McCall et al., which um, which is specifically more about Germanic, and it has its own methodology. Although the methodology isn't a big part of it. And then, oops, this is not the right one. Um, okay, sorry. I think I said was correct. I grabbed the wrong link. Um,
Yeah, steppe ancestry in Western Eurasia and the spread of the Germanic languages.
Yeah. And that one is like very very much about Germanic languages, and probably is more interesting to people who are especially don't have a background in it, because it kind of goes into more detail about that sort of thing. And then the other one is sort of migration era, so very similar, also very focused on Germanic, like spread of Germanic peoples and things. But I kind of like a little bit more methodologically. But they both have interesting methodology, and did I paste this? No, I didn't paste the same one. Sorry, the doors are just no. Did I? No, I didn't. Sorry, the doors are just so similar. I thought I did for a second. But yeah. So then the second one is yeah. But they both are very similar. They both have very interesting methodology, and they both have very interesting um uh uh um like finding and results. I hope I'm not alienating anyone or making anyone upset when I say this, but I think if you are like specifically the authors, I don't know if the authors are ever going to watch this, but I hope I'm not alienating them. But I think the McCall one has more interesting answers and kind of more interesting stories. So I think if you're not an expert, it's probably more interesting for you. But I think the methodology is a little bit more interesting for Spiel et al. And so I think if you want to get into the nitty gritty of the methodology, I would recommend that one. Both great papers, both have great methodology, both have great everything. So yeah, I recommend reading them basically.
Okay, the abstracts are certainly very interesting. Uh, with the the Goths as having Swedish ancestry. That makes some sense. But then interesting to suggest the Ostrogoths and Visigoths are predominantly of Southern European ancestry, implying the adoption of Gothic culture. Quote. That's interesting.
Yeah, yeah. I think that was a find that I'm not sure, but I know at least the like the Vielbark culture, like the Polish culture that was like debated to actually be Scandinavian in origin or not. But that's like a common finding of theirs. I think they both find the same thing of the Southern European ancestry as well. But I remember that like there's a lot of commonality. I mean, these papers came out like within a couple of weeks of each other, and are very similar topics basically. So um anyway, yeah, the golf thing was quite interesting. Um, yeah. Anyway, I'm happy to take any questions people have and do examples if people want them. I can share a whiteboard. Um, let's see who can start sharing.
How are people feeling in the chat?
Oh, I didn't even see the chat. Um, I'm glad I was nice, Matt, and SPSS. I'm not sure what SPSS is, to be totally honest. But yeah, if people in the chat are participants are wanting to do any questions, please throw them at me. I'm like, as you can tell, I'm very excited to talk about stuff. I could talk about it all day. It's like a joy to have a captive audience that wants to listen to me bore them about it. Because normally I start ranting about it. People are like, oh, Daniel, like, please change channel. Because
I love. I I think that there's a huge number of people who are interested in this kind of thing, and actually have you lay out the methodology behind it. I think is so important uh, because I think part of what gets lost in the noise when things like this are discussed in public fora is just like, well, how do people come to these conclusions, right? What is it that you're looking at? And it's obviously a lot of number work.
Yeah, I very much agree, and I mean I have much less, I don't know, life experience on like what is not important in academia and science and whatnot. But I feel like I feel like people I feel like it's important to like educate people. I think it's a very important, like, this whole point of science, in my opinion, is to spread knowledge. And I think that like an important thing of if you want to learn these sorts of things as like a just kind of random person is to read the papers. Even though I think pop science is great, I think it's always better just to read the actual sources or whatever.
Mhm.
And so hoping that what I've been doing is giving people a way of at least somewhat reading the paper and kind of critiquing it, or not even not critiquing it, at least understanding why they come to the conclusion they come to, right? So you can read some sort of F3 and F4 statistic. Although actually, like I said, these two papers I posted don't really use them as much because they're comparing such closely related populations. They kind of break down. They might use them a little bit, but anyway. Although actually, Spiegel et al. His methodology is based on F2, F3, F4 stuff. So
Okay.
um, it's kind of merging of like the kind of mathematical framework of F2 statistics with haplotype based methodologies, which is again, I think it's a pretty cool idea. Anyway, but like in general, if you read an ancient DNA paper, if you read like some of like the like Lazarides 2016 or whatever, which is one of the big like into or I think it's Allentoff's like 2015 is the other one. I mean, there's Hock et al. 2015. Anyway, there's a few of these big papers that are about Indo-European languages that people probably find interesting to read, and hopefully this will give you a way of reading them basically, and then like seeing, oh, this is why they're saying that these populations are related.
Hmm. The real test of this is going to be reading one of these and seeing if I understand it better now, which I suspect I will.
Yeah,
Uh,
I hope so. But like I said, these papers, unfortunately, I mean, I wanted to give them to you guys because I think they're the most like Jackson Crawford papers, especially recently, but they actually use less of these statistics. But
well, still understanding like I said, what goes into it a little bit better. I think we should probably call it right now just to make sure this doesn't go too long, because again, people get scared away by long run times.
no,
Uh,
don't worry, that's fine for me.
unless there are other direct questions. I've really appreciated your time and the way that you've explained this. Again, like I've had to think pretty hard, but I think that I followed you, and I'm going to see if I can read one of these papers with better understanding now. So I really appreciate the opportunity for that.
Perfect. Yeah, let me know, and thank you again for having me on. I think it's an important part of, even though I'm a very like starting my science career. I think I think it's the point of science is to share knowledge, and I also think that yeah, I just love talking about this. So thank you for giving me the opportunity to do so, because
Well,
it's
very much agree about the point of science. Enjoyed listening to you, and well, for now. Wish you all the best and all the best to everybody else.